So we're certainly witnessing some very exciting developments in the field of cancer immunotherapy with the continual string of FDA approvals of new immunotherapeutic agents for an increasing number of cancer types. We've seen some high-profile success stories like former President Jimmy Carter being completely cleared of melanoma that spread to his brain. But we also see the limitations of the latest immunotherapies as well. Not every patient benefits, and complete cures are relatively rare. But it's these limitations that also provide guidance for current laboratory research in discovering and developing next-generation immunotherapies that improve upon the latest advances. So the most pivotal advance in the field could arguably be traced back to 2011 when the FDA approved the first of an entirely new class of cancer drugs called immune checkpoint blockers, starting with ipilimumab, a CTLA-4 blocker for melanoma. And then came along a second class of immune checkpoint blockers with even more effectiveness called the PD-1 and PD-L1 blockers, starting with pembrolizumab and nivolumab for melanoma. These approvals were then extended to certain cases of lung cancer, kidney cancer, Hodgkin's lymphoma, a new drug called atezolizumab, the first PD-L1 blocker approved for bladder cancer earlier this year, and even the first combination immunotherapy consisting of nivolumab and ipilimumab approved last year for melanoma. Currently, there are over 700 registered clinical trials for PD-1 or PD-L1 blockers for a broad range of cancer types. At least 10 of those trials involve mesothelioma, and this is astonishing because exactly 10 years ago, this month, September of 2006, I was presenting my doctoral dissertation on nivolumab PD-1 immunotherapy at the UC Norris Cancer Center, and at the time we thought we were just working on melanoma. We thought that it would just be a melanoma-specific drug, and now a decade later, it's very encouraging, certainly, to see that the advances in melanoma have now trickled over into other cancers, potentially even proving to be an effective treatment option for mesothelioma. So PD-1 and PD-L1 blockers are engineered antibodies that block specific inhibitory receptors of the immune system, namely programmed death 1 receptor and one of its counterparts, programmed death ligand 1. These inhibitory molecules exist naturally in the body, primarily to prevent the immune system from attacking normal healthy tissue, but it turns out that tumors exploit these processes as well to shield itself from the immune system, and by blocking PD-1 or PD-L1, we can at least partially break down this defensive shield and allow T cells of the immune system to mount an attack on tumor cells. As far as the clinical effects of PD-1 and PD-L1 blockers, the primary effect is extended overall survival, a decreased chance of death. There's measurable tumor shrinkage in select cases, typically on the lower end, around 15 to 25% of treated patients. For most cancers, achieving complete remission is rarer, with the notable exception of maybe melanoma. The combination of nivolumab plus ipilimumab has been shown to induce complete responses in about 10 to 20% patients, and now this combination is now also being tested in pleural mesothelioma. Responding patients can experience durable benefit quite often. That can sometimes last years even after stopping treatment. The clinical data for pleural meso is still somewhat limited and somewhat immature, although the most recent publicly available data show a response rate of about 28% to pembrolizumab, but only a response rate of 9.4% to avelumab, which is a new experimental PD-L1 blocker. So the responding patients experience only partial tumor shrinkage with no complete responses, with the exception of maybe one of Terry Jahan's patients that he discussed earlier. But for the most part, we're only seeing partial tumor regressions, no complete responses. And this is what I meant earlier about the limitations of the latest immunotherapies. They don't benefit every patient, and complete responses are exceedingly rare for most cancers. On the bright side, we're now starting to learn more about potential drug resistance mechanisms. For instance, in melanoma patients, the use of PD-1 blockers can induce T-cell mediated inflammation in the tumor microenvironment, but this also induces compensatory upregulation of other inhibitory molecules, such as more PD-L1 being expressed on tumor tissue, which can interact with a second receptor called CD80 on activated T cells. This interaction results in downregulation of T cell activity. This is what we call adaptive immune resistance, and there are animal tumor studies now that suggest that the use of PD-1 blockers can also induce compensatory upregulation of yet another inhibitory receptor called TIM3, which has multiple ligands, including galectin-9, on tumor tissue. And this interaction also shuts down T cells. So you can see that the balance of the immune system versus tumors is very dynamic and regulated by multiple inhibitory mechanisms. So the logical progression of immunotherapy is likely the combinatorial use of multiple drugs that can block or otherwise overcome the effects of these multiple inhibitory pathways. And there is now a notable effort in clinical trials combining different combinations of immunotherapies. The combination of a PD-1 blocker with a CTLA-4 blocker has already been proven to be more effective in treating melanoma than either drug class alone. The combination of a PD-1 blocker with a PD-L1 blocker or a TIM-3 blocker seems like a rational combination to combat adaptive immune resistance for future clinical trials. Then there's a host of other immunotherapies that have potential to synergize with PD-1 or PD-L1 blockers. I'll categorically call them biological response modifiers since they encompass a broad range of immunotherapies that have different mechanisms of action. So the biological response modifiers that are most likely relevant for enhancing the effectiveness of existing immunotherapies like PD-1 or PD-L1 blockers likely involve those that directly modulate T cell activity within the tumor tissue, such as those agents that promote T cell trafficking or influx to tumors, so getting more T cells into tumor tissue. These we generally call chemokines, and those agents that promote T cell survival and effector functions such as certain cytokines. Now, getting chemokines and cytokines delivered into tumor tissue in sufficient amounts to have an effect is a bit of a challenge from a technical standpoint. Some approaches utilize novel fusion proteins where chemokines or cytokines are linked to other proteins that can carry them in to tumor tissue. There's nanoparticle technology for that purpose as well. Both of these areas are outside the scope of our research. What our lab has been focusing on and exploring is the use of a type of adult stem cell called mesenchymal stem cells that have natural tumor homing properties and quite possibly might also be universally compatible between different patients with different genetic backgrounds. So these mesenchymal stem cells might be suitable carriers for delivering immunotherapeutic agents into tumor tissue. So mesenchymal stem cells are naturally occurring cells in the body. They primarily serve as a reservoir to maintain or regenerate tissue, primarily fat, cartilage, and bone, possibly also muscle, neurons, and blood vessels. So there's a lot of interest in these cells for regenerative medicine purposes. For therapeutic purposes, they can be harvested from different tissue types, such as bone marrow, body fat, cord blood, and term placentas. We work primarily with bone marrow and term placentas, since those are the more easily procured tissues due to our association with UCLA Ronald Reagan Hospital. The earliest reports of using mesenchymal stem cells to treat experimental tumors in animals goes back to at least the early 2000s, so well over 10 years now. The study most relevant for pleural meso was published a couple of years ago by a group in the UK where human mesothelioma cells were implanted into the chest cavities of mice, and then the mice were treated with engineered mesenchymal stem cells engineered to produce a tumor cytotoxic protein called Pleural. And this resulted in significant inhibition in the growth of tumors in the chest cavity. So in this particular study, it was interesting that intravenous infusion of the therapy was more effective than intrapleural infusion. Whether or not that holds true will have to be determined later in clinical trials, but nonetheless, it's studies like these that offer the intriguing possibility that mesenchymal stem cells can also be engineered to deliver immunotherapeutic agents. So the first step towards developing such therapies involves laboratory studies with human tissue to demonstrate proof of concept that these cells can indeed be engineered for immunotherapeutic purposes. The production of mesenchymal stem cells, regardless of the tissue source, goes through the same general process. There's an initial digestion or separation phase to break down the original tissue source. After several weeks of growth and expansion and clearing out unwanted cells, we're left with a population of purified mesenchymal stem cells, which can then be used immediately or frozen away for future use. This is the step where we begin the engineering process to repurpose these cells for immunotherapeutic purposes. We use viruses to insert immune genes into these mesenchymal stem cells. We've started out with genes encoding for interferon alpha and IL-12, two cytokines that have well-defined immunotherapeutic properties. We screen these prototype preparations for production of these cytokines and also test their ability to enhance or boost human T cell activity in vitro. And thus far, it appears that the IL-12 producing mesenchymal stem cells have the best capacity for enhancing human T cell activity in vitro compared to controls on the right. So yes, we can indeed engineer these cells with immune genes that have immune boosting properties. Now, interestingly enough, and this was a bit of a surprise to us, the interferon alpha producing mesenchymal stem cells had actually enhanced immunosuppressive activity relative to controls. And this might be of interest to autoimmunity experts, for instance, who are looking for novel ways to shut down immune responses, but for the purposes of treating cancer, we need to do the opposite. We need to boost immune responses, specifically T cells. And thus far, it appears that IL-12 producing mesenchymal stem cells might be suitable for that purpose. Another appealing characteristic of IL-12 producing mesenchymal stem cells is that IL-12 does not appear to significantly alter the secretome of these cells. We can get these cells to produce large amounts of IL-12 while keeping at least 37 other naturally secreted proteins near baseline levels. Interferon alpha, on the other hand, results in significant changes in at least 7 or 8 other naturally secreted proteins. So we're not sure about what the physiological significance of this would be, but it could partially explain their enhanced immunosuppressive activity in our laboratory tests. But nonetheless, it does appear that IL-12 is a real promising foundation for engineering these cells with additional immunotherapeutic components on top of IL-12. As far as being a theoretical treatment for pleural mesothelioma, IL-12 secreting mesenchymal stem cells might be useful for treating residual disease remaining after surgery. So after pleurectomy decortication surgery, the chest tube drainage specimens have undetectable levels of IL-12 in the days following surgery. So the chest cavity appears deficient in a protein that is highly supportive of T cell activity. So the use of mesenchymal stem cells or other specially designed drug carriers to release IL-12 in the chest cavity, it seems like a rational strategy to treat residual disease after surgery if used as a standalone therapy or potentially in combination with existing immunotherapies like PD-1 or PD-L1 blockers. So there are additional immunotherapeutic components that we could potentially insert into these genes as well on top of IL-12. Other T cell activating cytokines such as IL-2 or IL-15, T cell trafficking molecules such as LIGHT or certain members of the CCL or CXCL chemokine families, and intriguingly, certain proteins that have now been identified as having high potency for blocking certain immune checkpoints such as soluble CD80 or high affinity PD-1 variants. Multiple components on this track could theoretically be combined into a single mesenchymal stem cell product in addition to safety components such as suicide genes, which would allow us a mechanism to quickly destroy these cells in the event of serious side effects. And I think over the long term, the inclusion of suicide genes will improve the overall clinical safety profile of all cell-based immunotherapies. So key points to summarize, immunotherapy is rapidly moving forward as a new option for treating diverse cancers. It appears that the PD-1 and PD-L1 immune checkpoint blockers look like the centerpiece for combination immunotherapies moving forward. Despite the recent advances, there's still a significant percentage of patients that are unresponsive to immune checkpoint blockers, and achieving complete remission is relatively rare. And we believe now that countering adaptive immune resistance through combination immunotherapy is likely the key to improve tumor responses. And mesenchymal stem cells might be suitable vectors for countering this immune resistance in the tumor microenvironment through their ability to deliver immunotherapeutic agents specifically into tumor tissue. So lastly, acknowledgements. Irina Yankulescu, who helped me launch the Pacific Mesothelioma Center Lab several years ago and get it operational by 2013. She's since retired from bench work to become a writer over at Cedars-Sinai, so best of luck to her, of course. The lab has now evolved into one where we have a steady supply of students coming through from local universities now, namely UCLA and Loyola Marymount. I get emails almost every day now from students wanting to work in the lab, so I guess they've really caught on to the immunotherapy bandwagon as well, even though that they're undergraduates. Our Scientific advisor Robert Cameron, and research funding provided primarily by the Pacific Heart, Lung, and Blood Institute, and grants from the Richard Schultz Foundation, the H.N. and Francis Berger Foundation, and the Kazane McLean Foundation. Thank you.